Okay, today we will be studying about a chapter in your syllabus, that is histology and anatomy of the flowering plants. In histology and anatomy of the flowering plants, we will be studying about what is tissue, what is the definition of tissue, what is cell, what are the different types of tissue, what characterizations is there, tissue is there if it is a simple tissue, permanent tissue, and if it is a simple tissue, what type of tissues are there, what is the structure, what is the main function of the particular tissue in the growth of the plant. Okay. In that regard, we will be following first with the definition. As we said, it is a histology and anatomy of flowering plants. What is histology actually? Histology is nothing but it is a definition like it is a study, detailed study of internal structure of the tissues as revealed under the microscope. When we see study of tissues is called histology. Now, what is anatomy? Anatomy is the detailed internal structure of a plant as we take the TS, transverse section of a tissue in the under the microscope and see, then that type of internal structure. When we see the transverse section of a tissue, we will see the internal structure, the arrangement of the epidermis, hypodermis, cortex, xylem, phloem and pith, all that internal structure of a stem, root or leaf. That detailed study is called as anatomy of a particular plant tissue, right. Now, before going all this, we will go into we all know that cell. What is cell? Cell is considered as the basic structural and functional unit of a body or an organism. So, cell, a body is made up of group of cells, and these cells will make, group of cells will make into tissues, and tissues will make into body organs or plant organs, and all these organs will together make into a system or total plant body, right. So, here we can say that cell is considered as the basic functional unit, and from there a group of cells will make together a tissue, and group of tissues will make an organ. Like we have branches, stems, because they are combination of tissues, and from there these all organs will club together and make a total plant body. Okay. So, this is the definition of tissue, histology and anatomy. So, tissue is nothing but a group of cells put together and organized into a different organs so that it functions. Now, when we come to tissues chapter, tissues are basically of two types. Those are meristematic tissue and permanent tissues. Now, these are the classified tissues. Meristematic tissues are those tissues. This classification is based on the basis of the cell division, the capacity of cell division. If a tissue is undergoing a capacity of meristematic tissue and permanent tissue, the division of the tissue is done on the basis or the capacity of the cell division in a particular tissue. If the tissue is having the total capacity of cell division, then that is called as meristematic tissue, and if it does not have or it does not retain the capacity of cell division, then that is called as permanent tissue, right. Now, meristematic tissue are again divided into two, primary and secondary meristems, and again this primary meristem, on the basis of positioning, it is position in the plant body where it is present, they are divided into apical meristem, lateral meristem and intercalary meristem. So, now before going, and again permanent tissues are of three types: simple tissues, complex tissues and secretory tissues, special tissues. These all we will study in detail later. Simple tissues, as you all know, there are three types: parenchyma, collenchyma and sclerenchyma. Complex tissues, we have xylem and phloem, and again in xylem it is we are supposed to study about the trachea structure and vessel structure. Phloem, we are studying about sieve cells and sieve tubes. In special tissues, like in some plants, the plant does not take inside the excess of material which is not required to the plant. Excess of material which is not needed to the plant is excreted out or secreted out, then that is done by some special glands or cells. Those are called as secretory cells and secretory glands. Okay. Now, first we will come to before starting with the tissue, first we will go with the meristematic tissue. In meristematic tissue, before entering into detailed study of meristematic tissue, let us first consider the characteristic features. Why we consider it as the meristematic tissue? What is the need to study in detail about meristematic tissue? Now, when we are studying in detail meristematic tissue, the first and the foremost point we have is it has a continuous state of cell division. These are in a continuous state of cell division, and again these cells may be round, अगेन फिर ड्रॉ करना. Okay. Now first we'll study about meristematic tissues. Now, what are the first basic characteristics why we are studying meristematic tissue is, it has, meristematic tissues are those tissues which has the power of cell division, and when we come, these cells are normally comprising of, these may be round in shape, they may be oval in shape, and these cells will have less number of intercellular spaces, or there will be no intercellular spaces present in them. These are living cells, and these do not they are helpful in the storing of reserve food material. And again, as we said, meristematic tissue is again of two types, primary meristems and secondary meristems. Right? Now, what is primary meristem? Primary meristem is the one which is formed from the primary plant body, is called as primary meristem. Okay. Now, in this one bit is there, one thing is that this meristematic tissue forms the basic of all the ground tissues of the plant. Any part of the plant body where there is a state of continuous cell division, there you can say that you are having a meristematic tissue. Now, in this you are having the division of the meristematic tissue will be equal, it will be depending upon the position from where this meristematic tissue is growing, either it may be anticlinal or periclinal. When it is a process of division, so for example, in case of cells of dermatogen, the tissues where the dermatogen layer is developing, it will develop in anticlinal division will be there. What is anticlinal division? The division will be at right angles to that of the structure which is developing. Now, in case of cambial tissue, cambial cells or cambial tissue, cambial cells, the division of the meristematic tissue division will be parallel to the surface. Right? So, now here depending upon the positions, meristematic tissues have the division like anticlinal and periclinal division. Anticlinal division example is in the case of meristematic tissue shows anticlinal type of division in the case of the cells where dermatogen layer is developed, and cambial cells will show the division of the cells parallel to the surface. Now, primary meristems are those. Now, when we come to the types of meristems, there are, as we said earlier, okay, as we said that there is meristematic tissue is of two types. First, we will be studying about the primary meristems. Okay. Now, in my primary meristems, you what as the name itself indicates, primary means it is the tissue which was developed in the primary plant body, or it is a tissue which is developed from the embryonic part of the body, embryonic stage of the plant, and this develops in the primary plant body. Now, here this, in case of some as you are given examples in your syllabus, in case of palms and banana plant. Okay, in this type of monocots, where these are monocots, in this, when there is no secondary growth. Now, when there is no secondary growth is occurring, we need the plant to grow in the length. Okay, so what we'll do, the apical cells which are helpful will help in the multiplication and movement, adding more to the apical initials which are present at the tip of the plants. Will have these primary meristematic tissue present at the apical initials, which will help in the multiplication and increase in the height of the plant body. Okay. Now, this type of cells which are help in the embryonical growth or in the initial stage of multiplication of the plant body, then those types of tissues are seen in the examples like palms and banana plant. Okay. Now, next, these type of tissues which helps in the initial growth increase in the height of the initial growth of the tissues, those are also called as primary thickening meristems. Means what they do here they will be helpful in multiplication where there is no secondary growth. They will go towards the initial stages, apical initials. They will multiply the cells and they will help in the increasing the thickness of the tissue. Then those are called as primary thickening meristems. Okay, no? Next, one more you are having. Sorry. Now, this comes. Now, when we come here, these primary meristems are again divided into 3 types. Now, this type of classification is based on the basis of position. If, for example, this is a diagram, here, depending on the position, you can have apical meristem, you can have lateral meristem, you can have intercalary meristems. Means, if it is a plant, depending on the topmost position, on the middle position and towards the sides of the plant, the growth of the tissues, meristematic tissues present in the primary plant body are categorized into apical, lateral and intercalary meristem. Now, apical meristems, these are the meristems which are present at the tip of the roots, stems and branches. These are the meristems which are present at the apical meristems, which are present at the apical initials or apical tips at the top of the root, stem and leaves. Sorry, stems and branches. Okay. Now, what is happening here? These initials, apical initials will undergo, again this apical meristems is having 2 more parts. If it is an apical term, here this is the apical meristem. So, here you have 2 sections. First one is called as promeristem and promeristem, and another one is a meristematic zone. So, apical meristems which are present almost at the tips of the roots, stems and branches. These type of apical meristems are divided into 2 zones. Those are called as promeristematic zone, promeristem and meristematic zone. So, here these promeristematic zones are group of division of cells will occur at the promeristem will show a group of cells with the group of initials, apical initial cells will be there. And again this meristematic zone will show the continuous division, multiplication of the cells, and this will increase in the height of the tip of the plant. Okay, next one, these meristematic zones are again divided into 3. What are those? These are divided into protoderm. It is again divided into procambium and again divided into ground meristem. Okay. So, here again I will repeat, meristematic tissue on the basis of position 3 types are there: apical, lateral, intercalary. First we are talking about apical meristem. In apical meristem, at the tip of all the roots, stems and branches you will see the presence of apical meristems. This apical meristem has 2 parts, those are called as promeristems and meristematic tissue. This promeristem has a group of division of cells at the initials, and this meristematic zones helps in the dividing of the cells, and this meristematic zone is again divided into 3 parts: protoderm, procambium and ground meristem. Now, what does this protoderm, procambium and ground meristem help in? Okay, protoderm, procambium and ground meristem. Protoderm will ultimately give rise to epidermal tissue system. This will give rise to primary vascular tissue system, and ground meristem, as we all know, it will give rise to remaining cortex and pith. Okay. Now, this is about apical meristem. Now, next type of meristematic tissue on the basis of the position is intercalary meristem. What is intercalary meristem? Intercalary, as the name indicates, inter means in between. These are the cells which are formed or intercalated in between the permanent tissues. Okay. So now this originate from the apical meristems. These are the tissues which originate from the apical meristem when their total growth is over, when their development, their portion gets totally detached from the original plant body. For example, in grasses, these are normally present in the internodal regions of the leaves. This may also helpful in the development of the flower or new branch from this region. Now next comes is the